Hi everyone, welcome to our Microbial Ecology video. Today our group will present about microbial ecology of estuarine system. So before we start with our exciting discussion, let me introduce these presenters to you guys. First is Anis Hanani, who will introduce about estuarine ecosystem, and she also will conclude the whole presentation in the end of this video. Next is Ainur Shuhada, who will present about mechanism of microbe in the estuaries. The third one is Nuraisha, who will expose to you about recent case studies related to estuarine ecosystem. And lastly is me, Aina Ahida, who will present to you guys about ecological impact of the interactions between plant, animal, and microbe. So let's start now.
First of all, let me briefly introduce the microbial ecology process of the estuarine ecosystem and its significance. Estuaries is the connecting link where fresh water from the land meets with salt water from the ocean. They are considered as dynamic and productive environments which support a diverse array of life forms. Human civilization relies on estuaries, and many estuarine ecosystem services are provided by microbial communities. High rates of primary production supporting the harvest of commercially valuable species through aquaculture and fisheries, the conversion of terrestrial and anthropogenic materials to help maintain the water quality, and mutualisms that maintain the accumulation and storage of blue carbon are some examples of these services. Microorganisms are central to estuarine ecosystems, inhabiting every niche and profoundly influencing crucial ecological processes. Microbial ecology of estuaries plays a crucial role in maintaining ecosystems' functionality. Sulfur and methane metabolism are important aspects of estuarine biogeochemical cycling, driving the critical processes which affect the availability of nutrients, emission of greenhouse gases, and ecosystem health. The dynamic interplay between these metabolisms are regulated by environmental factors such as oxygen gradients, sulfate concentration, and organic matter availability, which ensuring the stability and functionality of estuarine environments. In order to preserve estuarine ecosystems and minimize their influence on the climate globally, it is essential to comprehend and control these microbial processes.
Here are the microbial respiratory processes in sediments. Newly derived organic material at the surface of sediments is selectively decomposed, ultimately producing CO2, acetate, and H2 that can be used to support methanogenesis. The remaining organic material that is resistant to decomposition is stored as blue carbon. The methane that is produced via methanogenesis can then be reoxidized through microbial transformations using several electron acceptors including sulfate ion, nitric ion, and oxygen. So the first one is sulfur metabolism. The sources of biogenic sulfur compounds is derived from algae, phytoplankton, and estuarine macrophytes like Spiratina alterniflora. The role in climate. The compounds like dimethyl sulfide act as cloud condensation nuclei, influencing the climate system. So next is the microbial processes involved in these metabolisms, including sulfate reduction, sulfide oxidation, and syntrophy interactions. Sulfate reduction, predominant pathway for organic matter decomposition in coastal system despite lower energy yield compared to O2 and nitric ion. Sulfide oxidation is mediated by Proteobacteria and Chlorobia using oxygen and nitric ion as terminal electron acceptors. So for the syntrophy interactions, it is important for coupling sulfate reduction with methane oxidation. Moving on to the second one, which is methane metabolism. The sources and production. Methane produced via methanogenesis from organic substrates like methanol and acetate. The methane oxidation. It occurs mainly through anaerobic methane oxidation, which is methanotrophy, involving diverse bacterial and archaeal lineage. Next is microbiodiversity. Methanogens belong to archaea and perform methanogenesis under anaerobic conditions. The third one is interactions and environmental factors. For the competition and cooperation, methanogens and sulfate reducers compete for substrates but can coexist through syntrophic interactions. As for the environmental influence, the rates of sulfate reduction and methane production vary seasonally and across estuarine habitats. The last one is genetic and metabolic flexibility. The genetic diversity. Sulfate reduction can be carried out by multiple microbial phyla, while sulfur oxidation is more restricted to specific clades. Metabolic strategies. Unique strategies include nitrate ion storage in vacuoles such as Pegia toa and electron transfer through Cabal bacteria such as Desulfobulbaceae. As for the conclusion, this captures the key microbial mechanisms and interactions involved in sulfur and methane cycles within estuarine ecosystems.
Moving to the next part, which is a recent case study in microbial ecology of estuarine ecosystems. Recent case study on abolition dominates methane emission in stratified coastal waters. This study investigates methane dynamics in coastal environment focusing on the Tvrmin archipelago in the Baltic Sea. This area represents many estuarine systems with low salinity and high methane inventories. The research aims to understand the origins and processes regulating methane formation and oxidation in these coastal waters, which is crucial for assessing climate change feedbacks. Comparisons are made between methane diffusion and oxidation at nearshore and offshore stations, examining sediment biogeochemistry, microbiology, and isotopic signatures to determine the predominant sources and mechanism of methane emissions in this region. The study found that at the nearshore coastal station, methane diffusion from the sediment into the water column was negligible due to the effective oxidation within the upper sediment layer. In contrast, the offshore station exhibit the significant benthic methane diffusion, but most methane was oxidized before reaching the bottom water, and the thermocline acted as a barrier preventing methane from reaching surface waters. This finding suggests that abolition may play a critical role in methane emissions from the various stratified breakage coastal systems worldwide, potentially leading to an underestimation of global methane emissions.
Next, the ecological impact between plant microbe and animal microbe interactions have been proved to provide concrete benefits in several studies. For example, through nutrient provisioning, defense, and chemical signaling. However, in some cases, the disturbance from outer environment may lead to decreased fitness and disease. Hence, we should understand the specific nature of host microbe interactions and how they will respond to environmental change for the sustainability of estuarine habitats and commercially important organisms. First, we look into microbe-animal interactions. Animals habituated in estuaries rely heavily on microbiomes for a range of different processes, such as reproductive and settling cues for marine invertebrates. Microbes also can produce key nutrients and vitamins for their hosts. This interaction plays a vital role in marine systems, corals, and sponges. Here, I highlighted one of the most important microbe-animal associations for estuaries function, which is reef building bivalve. These bivalve couple pelagic and benthic systems by filtering phytoplankton from the water and shuttling waste to the sediments in the form of biodeposits. So what are biodeposits? Biodeposits are deposits from biological origin which can promote microbially mediated benthic denitrification. The filtration of phytoplankton from water may improve water quality and decrease the symptoms of eutrophication that is caused by excess nitrogen and phosphorus. The bioreactivity and denitrification potential of those deposits along with microbes that decompose them vary by bivalve species, which can be quite challenging to predict the Estuarine nitrogen removal. On the other hand, symbiotic relationship between benthic macrofauna and through their engineering sediments can lead to increased oxygen penetration and coupled nitrification-denitrification. Even though microbe-animal interactions pose several benefits to the ecosystems, however, this role of microbe may reverse depending on environmental conditions. Slight change in environment may shift the function of microbe, which can lead to spread of disease. The term of such situation is dysbiosis, which means a shift in microbiomes that is associated with disease can be induced by environmental change. Dysbiosis is very fatal as it is not only affect the estuarine ecosystem, it also may cause disease to commercially important estuarine organisms such as oysters and lobster. Animal host microbiomes are sensitive to both environmental change and stress-induced change in host physiology. Hence, we have to make sure maximum understanding on microbial shift that result from global change. Next is the ecological impact of microbe-plant interactions. We can see the interaction of microbes with seagrasses, submerged aquatic vegetation, salt marshes, and mangrove, where this interaction contribute to plant resistance towards disease as well as better growth of plant. For example, the interaction between nitrogen fixing and sulfur oxidizing with seagrass can be seen in the good quality of seagrass growth in oligotrophic waters, such as in the Mediterranean Sea. The release of oxygen from seagrass roots into sediments create oxic microzones, which leads to an increased abundance of sulfur oxidizing bacteria compared to those found in bulk sediments and decreased concentrations of toxic hydrogen sulfide. Recent genomic study has shown that sulfur oxidizing and nitrogen fixing microbes support the global distribution of seagrasses in estuarine and coastal waters. As with seagrass systems, the microbiomes of salt marshes and mangrove vegetation contribute to several critical ecosystem functions such as fungi, which is these fungi act as primary decomposers of standing dead biomass and are important in facilitating salt marsh denitrification. Rhizosphere also play a significant role for nitrogen fixation and continuous sulfur cycle in salt marsh and mangrove sediments. Lastly, the microbiome found on the blades of macroalgae provide defense, nutrient exchange, reproduction, and settlement to plants successive in salt marsh and mangrove swamp.
Before the end of this video, let me summarize the key points which can be a vital takeaway about the microbial ecology in the estuarine ecosystem. Interaction between sulfur and methane metabolisms in estuarine environments influenced by environmental factors, genetic adaptations, and metabolic flexibility of microbial communities. Based on a research paper which related to the topic, estuarine systems are characterized by low salinity and high methane inventories, highlighting their unique environmental conditions and the ecological processes associated with methane metabolism. Animal microbe and plant microbe mutualisms in estuaries are fundamental in maintaining the stability of a dynamic environment. Health and resilience of estuarine ecosystems depends on the continuous research on microbial diversity and function, given the immense roles that microorganisms play in estuaries processes. In conclusion, Embracing microbial ecology as a cornerstone of estuarine ecosystem management is essential for fostering sustainable practices which safeguard these vital habitats for future generations.